last stop on this brief tour of multicellularity is the red algae. A couple of facts about them. First of all, no cilia ever. In fact, their cells have no known motile mechanism. There's evidence from sequencing things that they once had cilia, that is, that ancestors of the red algae had cilia at some point. So this is a secondary loss. They've also lost centrioles. Okay. In general, there's some idea that they went through an evolutionary bottleneck. Many of the basally branching members of the red algae are unicellular extremophiles, things that live in hot springs, for example. And the red algae in general show some evidence of genome reduction, as if perhaps the group as we know it now was were all descended from some sort of extremophile branch of what was once a bigger part of the eukaryotic tree. The red algae make cell walls with cellulose plus other polysaccharides, including agarose, which people use for gels in the lab, and also things that you find perhaps unwelcome, thickening your ice cream or whipping cream or whatever it is, additives in foods to make them gooey. The red algal cell wall is gooier, intrinsically more of a gel, than the kind of stiff shells of, say, Chlamydomonas or land plants. Instead of the kinds of cell junctions that animal cells make, or the wall-to-wall fusions of other plants, red algal cells hang on to each other after division by making what are called pit plugs or pit connections. During the final phase of cytokinesis, the furrowing zone becomes densely packed with endomembrane sacs, which are secreting something. They're secreting a blob of stuff in there, which becomes a little rivet between the two daughter cells. This rivet does not appear to be a cytoplasmic connection, a means of communication, the way, say, plasmodesmata are in land plants. So these cells appear to be fully partitioned, but they are connected to each other by this kind of firm rivet that holds filaments together. Much of the growth of red algae, from simple forms to even large-bodied forms, is based on filaments extended perhaps by the proliferation of an apical cell, followed by expansion of cells subsequently, and they're all connected together by pit plugs like that, in addition to the cell wall materials, of course. A common theme in one of the common themes in red algal bodies is extensive use of cell fusion. Cell fusion, which we normally expect to see primarily between, say, gametes, is used in red algae as a means for one set of cells to nurture another for example. Cell fusion is also used to make secondary pit plugs. This is a primary pit plug. That is, it results from the division of a cell to make two daughters. I'm going to diagram two filaments developing here, side by side. Yes, I know, I left out one of the nuclei. Okay, so here we've got some filaments running alongside each other. Now, how are you going to make a big body? Excuse me, thallus. Thallus. Say thallus when you're around phycologists. How to make a big thallus out of a bunch of filaments? Red algae solve this problem by division, by an unequal division. So that's why I didn't draw a nucleus in here. This unequal division produces the following situation. It's going to take me a minute to draw, bear with me. This unequal division creates a tiny little cell connected to its sister by a pit plug. And the next thing that happens is that that little cell fuses with its big neighbor. Again, bear with me. The fusion of this tiny little cell with its neighbor injects a nucleus into the neighboring cell, leaving behind this pit plug as a connection between this now heterokaryotic cell and the cell which originally initiated this connection formation. Now, these aren't truly heterokaryotic if these are members of the same plant, derived from the same zygote, right? Nevertheless, this habit of injecting nuclei into each other, which is pervasive amongst red algae, what could possibly go wrong? Now, I should also mention in passing that very likely these are heavily multinucleate cells. Red algae use endoreplication and the production of many nuclei within a common cytoplasm to achieve large cell size. Within a red algal body, cell size might vary over several orders of magnitude, over as many as 4 orders of magnitude. Some of the cells get quite large, and not simply because of the expansion of a vacuole, but because of proliferation of nuclei within the cytoplasm. Many red algae pass through 3 distinct generations. A macroscopic so-called tetrasporophyte, which is likely to be an independent plant. You'll see what I mean by that. The tetrasporophyte is diploid, and as you can tell, it must bear spores. In addition, male and female gametophytes might also be free-living plants. Those will be haploid, of course, and bear gametes. They may have similar morphologies, that is, they may be isomorphic or very different. They may have heteromorphic alternation of generations, as in the green and brown algae. The third generation, the third body, the third multicellular body, is microscopic and parasitic carposporophyte. This third generation generally lives attached to and deriving nutrients from the female gametophyte in a way analogous to the growth of an embryo in a placenta. This is a diploid spore-bearing generation. In this case, the spores that are released by carposporophytes are diploid spores that will go on to form the tetrasporophyte. So the life cycle might look something like this. The tetrasporophyte some fraction of its cells undergo meiosis to produce tetraspores, so-called because very often they occur in groups of 4, as they ought to, where what you're seeing here is one pair that way and another pair that way. This is what they'll look like to people taking the lab if we manage to find them. Okay, each of these spores is a haploid cell. Remember, this is a diploid generation that goes off to make either a male or a female gametophyte. I'm surprised to know the male gametophyte bears male gametes, sperm, but for whatever reason, amongst red algae, the cells that are produced are always called spermatia. The female gametophyte bears what? No, it's not called an egg. It's called the carpogonium. And you can think of it sort of like a sessile attached gamete. That is, this cell, which can be fertilized by a spermatium, is attached to the female gametophyte. It's not like they cast out eggs into the water or something like that. So these fuse to make a diploid zygote of some sort. But things get a little weird in there. At any rate, the formation of this fusion product leads to the development of a little parasitic plant growing on the female gametophyte. That's the carposporophyte. It is also diploid, like the tetrasporophyte. The carposporophyte plant a large fraction of it goes to the production of carpospores, which are released. And each one can develop into a tetrasporophyte. The ones that I hope to be able to show in the lab within the tetrasporophyte, the tetraspores are formed in little packages in a plant that is otherwise strikingly similar to the gametophyte morphologically. So we will recognize them by the presence of tetraspores within the peripheral fronds. In contrast, the male gametophyte bears branches that develop large clusters of spermatia. They look very much like, I don't know, something like amaranth, the seed heads of amaranth. Well, what does it do with these? Well, assuredly it releases them. But here's the thing, no cilia. Remember, these are not things that will swim to a target. In fact, they are more like pollen in that sense. Gosh. Well, if they can't swim, what takes them to their targets? And what are their targets anyway? In the female gametophyte, amongst the various branches, the carpegonium has a little antenna. And if we blow up what this looks like, then along some filament within the plant, connected by pit plugs here, and perhaps including many nuclei per cell, a fertile branch ends with a carpegonium That's what that is. And this thing here is called a trichogyne. It's a kind of an antenna to collect the spermatium. Not unlike, if you will, the silk on an ear of corn. So this thing attached to a supporting cell down there. When a sperm binds, as it happens, there are two nuclei in the spermatium, I think, and it fuses with the trichogyne, and one nucleus moves down and one nucleus moves up. And eventually, once one of the male nuclei gets together with the carpogonial nucleus, a division separates the trichogyne. I remember reading somewhere an explanation for why they do it this way, which I thought was pretty interesting, but I've forgotten it. So I'll try and look it up before we have a question time if anyone wants to have an easy question to pose during question time. The answer might be, I still don't know, but we'll see. Anyway, so this thing, this product of fertilization, naturally is going to go on to form the carposporophyte. And it could do so directly. It could just attach to its supporting cell, start to develop branches. That ultimately become sufficient to produce, okay, here's the abandoned trichogyne here. So all these diploid things could eventually produce carpospores. And this here would be the carposporophyte. But they just couldn't stop with something simple and easy to understand like that. As it happens, what commonly takes place in the more derived forms of red algae, including the kind that I hope to be able to demonstrate to students in the lab, is that this cell then fuses with the supporting cells, and I'm not going to try and draw this. I don't understand it well enough myself to draw it. This cell fuses with some of the supporting cells, injecting this diploid nucleus, which it might have made several copies of in the meantime, into supporting cells and into many such cells, thus taking over and converting some fraction of the gametophyte tissue into carposporophyte producing tissue. And that's the sense in which the carposporophyte seems especially like a parasite. Remember, the female gametophyte is haploid, and the product of fertilization is diploid, and half of the genome in that diploid comes from some other individual, from some male gametophyte. So this cell is put in the position of accepting a different genome, a variant genome, into it by cell fusion. Of course, I mentioned these cells are already in the habit of fusing with each other, injecting nuclei into each other for the sake of making big bodies, fusing filaments together. What could possibly go wrong with this strategy? Well, for red algae, the big liability is perhaps not cancer, but parasitism. Deploying this strategy ubiquitously as they do, this habit of injecting nuclei into each other, and the need to tolerate nuclei from a different source as part of their reproductive strategy, within the red algae, parasites have arisen perhaps hundreds of times that take advantage of this to fuse with their host, inject nuclei, and take over host tissue, feeding on them and using their resources for their own benefit. Again, I hope to be able to find some years I find some and can bring them to the lab. Many of the red algal parasites are colorless. They don't have their own plastids, and they form little white plaques on the bodies of filamentous red algae. So they're fairly easy to recognize. Commonly, although not always, commonly, the parasite is a relatively close relative. That is, it's a sister. The parasite comes from a sister lineage to the host, so-called adelpho parasites. Adelpho for brother. The presumption is that this parasitism is pre-adapted by the habit of using cell fusion as part of the reproductive life history.